Okay, well, in this chapter 3, we're going to be studying chromosomes and how these things are involved in the, heredity. Okay, and we'll give a better explanation of what they are. One thing to keep in mind here is that before we could actually, when Mendel was doing his work, people were trying to figure out what the genetic material was. And with Mendel's work and some of the other stuff that was being done in cytology and biochemistry, they were able to establish the chromosome theory of heredity. So, what we're going to look into is that concept and what the chromosomes are. So, keep in mind that where the chromosomes go, that's where the genes go. Okay, so the genes are on chromosomes. So, chromosomes, the general structure of it is DNA, but it also has protein. So the protein helps to organize the DNA and compact it down so that it can be put into a very small nucleus. So we're going to look at chromosome transmission during cell division and in sexual reproduction. So here you can see the little blue colored, fluorescently colored chromosomes here. And then you can see the centrioles at each end producing the spindle for the separation of the chromosomes. Now, before we get started, I kind of need to introduce you to something. Because of the Human Genome Project and because of the amount of new types of diagnostic tools that we have developed as part of that result of the Human Genome Project, we are right now delving into the structure of the chromosome. Previously, we had not been able to figure that out, but because of our extra probes that we've created, because of the Human Genome Project, we are right now getting to a point to where we're starting to see how these chromosomes are organized in the nucleus so that genes can be selected. Now, based upon that, they're seeing, when they look in there, they're seeing that there is an organization of how these systems are set up, and that some are going to be some chromosomes are going to be basically collapsed down, concentrated, so that they won't be used for expression. Some are going to be opened up, so that it collapsed down as heterochromatin. The part that's opened up is euchromatin. And here's the thing: for all the different cell types in the human body, there's going to be different areas that are heterochromatin and different areas that are euchromatin. Not completely different, but different. Okay. And so one of the things they're starting to see is they're looking at this thing called a fractal globule. So if you take away the cell membrane, this is how we think the chromosomes might be organized. And this is the one right now, sort of the accepted particular one. There's another one called the equilibrium globule, where the chromosomes are just evenly mixed around here. But this seems to have each individual chromosome has its own particular zone within this globular structure. And so they're kind of thinking that this could be the concept of this. This was presented in 2009, and they're doing even more work in this particular field. But I just wanted you to be aware of how these things are going now, and there's going to be a lot of changes as we move along. It'll be interesting to figure out, based upon how this is organized, if chromosome structures on one chromosome can have an influence of gene expression on another chromosome. That'll be an interesting concept to see if we can figure that out. So let's get into chromosomes, okay? So we're going to be looking at eukaryotic chromosomes, and we'll take a look at prokaryotic chromosomes. But what we have now, what we dealt with at the beginning of the 20th century, we came up with this, the chromosome theory of inheritance, that Mendelian factors are genes, and these genes are located on chromosomes, okay? The number of chromosomes, with rare possible exceptions, is characteristic of the species. In mammals, we might have the same amount of DNA, but we might have different numbers of chromosomes. Our closest relative in the mammalians is the chimpanzee, and they have an extra set of chromosomes, which, by the way, is actually for us their two extra chromosomes are actually fused together to create our chromosome number 2. So we refer to that two extra chromosomes as chromosome 2A and 2B. We've actually sequenced them, and we can show that our chromosome 2 is a combination of those two chromosomes. So there's variations that have occurred out there. So let's introduce this chapter. Basically is a survey of reproduction at cellular level. So we'll look at the chromosomes. You can see here, this is a scanning electron microscope of a set of chromosomes that were taken at metaphase. We'll look at how the microscopic level, we'll look how this gives us information about the inheritance patterns, because at the microscopic level that helped us to understand the chromosome theory of inheritance. When we combine that cytology with Mendelian genetics, then we'll look at how it was determined how chromosomes are involved in the inheritance of traits. So these are going to be structures that we find in living cells that contain the genetic material. They contain the genes, the DNA sequences. So biochemically, chromosomes are composed of DNA, deoxynucleic acid, which is the genetic material in eukaryote for us, proteins, which provide an organized structure. So in eukaryotic, the DNA protein complex, the combination of these is called a chromatin. Okay, so the average diameter of a nucleus is about 6 micrometers, which occupies about 10% of the total cell volume. That's a very small amount, considering the fact that if we put all the DNA end to end from one single cell, it would actually be almost 2 meters in length. That's a lot of DNA to put inside of an area of only 6 micrometers. Remember, a micrometer is 1/1,000,000th the length of a meter. So what does that mean? How do we do this? How does that organize? Well, that's where the protein comes into play. The protein helps to reduce the overall size of this very tiny strand of DNA, and it helps to reduce the size of it so that it can be compacted down into this small nucleus so that it can also be utilized when certain genes need to be turned on. We'll do another study chapter on that as well. But first, let's go ahead and go into the distinctive cellular differentiations differences between two cell types, your prokaryotic cell types and your eukaryotic cell types. So in prokaryotes, they do not contain a nucleus. They have a region, and that means they don't have a membrane, basically. They do have a region that has a DNA in it. They refer to that as a nucleoid, okay? And that region is located in the cytoplasm. So the DNA is in direct contact with the cytoplasm. So if you're expressing a gene or having transcription, as soon as that transcript starts to come off, a ribosome will actually bind right to it. So you could actually have transcription and translation going on at the same time off of a messenger RNA. The cytoplasm is enclosed by a plasma membrane, which helps to regulate the nutritional uptake and waste excretion. Outside the membrane is a very rigid cell wall that protects it from breaking in the various environments that it's going to be exposed to. Some species contain other things like another outer membrane or a flagellum. So this is your prokaryotic. Here's an E. coli representation of this particular one. It's about 1 micron and about 1 micron in diameter, so only a couple of microns in length. When we compare this to a eukaryote, we have a lot more complex structure. So here's a 6 micrometer structure over here. This is the nucleus. It is a membrane-bound structure. In fact, it has 2 membranes. The outer membrane is actually associated with the endoplasmic reticulum. So the actual lumen of the endoplasmic reticulum is connected to the space between the 2 nuclear membranes. So it has membrane-bound organelles like mitochondria, and they also have 2 membranes. In plants, you also have the chloroplast in those. But it also has lysosomes, which basically play a role breaking down macromolecules that are no longer needed, or even the mitochondria. And a Golgi apparatus that is involved in modifications of proteins as well as trafficking of the proteins. So you generate the proteins there. They have to the proteins then, if they have to go to the nucleus, there's a signal that takes them back there, or if it goes to mitochondria or the lysosome, there's signals on those proteins that tell them to go that direction. So the work that's been done here is in the field of cytogenetics. And so in this field, it's a combination of a microscopic examination of chromosomes and the genetics, Mendelian genetics. So they look at the chromosomal composition of a particular cell or organism. And this allows us to look for individuals with abnormal chromosome numbers or structures, which gives us a little bit of hint of what's going on. And this all provides a way to distinguish between 2 closely related species.